Assalamualaikum and hello. Today our group will present about the mini project, and the title for the mini project is Plant Disease and its Management. So our group have decided to choose rye blast disease caused by Megnaphate griseae. Okay, and now I will explain about Megnaphate griseae. Okay, what is Megnaphate griseae? Okay, Megnaphate griseae is type pathogen of fungi that cause disease for the paddy plant, and the familiar disease that caused by the pathogen is rice blast disease. Okay, and this disease, and this pathogen known now is a cryptid species complex containing at least two non-interbreeding biological species with distinct genetic differences, and also some individual confined from the Digitara have been depicted as Megnaphate griseae. Okay, the remaining individual of the disconnected rice complex and number of other were called Megnaphate oryzae. So for this part has do some confusion, which is Megnaphate oryzae and Megnaphate griseae. So the journalist decided to using the both name to do article. And the Megnaphate griseae complex can moreover 10 other horticulture really critical cereal contain wheat, rye, grain, and pear millet, causing infection called impact melody or cause melody. Rust impact cause financially noteworthy 3 misfortune every year. Each year, it is assessed to devastate sufficient rice to nourish more than 60 million individuals, and the organism is known to happen in 85 nations worldwide. Now I will explain about cycle of Megnaphate griseae. Okay, for this part, our group have decided to choose only one cycle because the life cycle and disease cycle for this pathogen are related to each other, and also this pathogen are asexual, and and cycle of asexual for this pathogen cause infection to the plant. So our group have decided to choose one cycle for this pathogen. Okay, now I will explain the first stage. Okay, the first stage is attachment. Okay, attachment is the first step of infection by pathogen. In Megnaphate griseae, the adhesive material known as the spore tip is preformed and stored in the periplasmic region of the apical conidial cell. This cell is released upon contact with plant or artificial surface and form a viscous port to attaching the conidium to the substrate. Okay, after done attaching to the surface, this conidia will germination, and this part we call germination. Okay, after attached, conidia germinate and produce germination tube or germ tube. Okay, for conidia attached to solid surface, nutrients are not required for germination, and single germ tube usually emerge from apical or basal cell of the conidia. Okay, after germinate and produce germination, this germination tube will produce Oprosorium. Okay, how Oprosorium produce? Okay, now we explain. Okay, this part we call Oprosorium, and the germ tube of this pathogen recognize the gut cell lip to form Oprosoria directly over stomata. Okay, this pathogen normally penetrate rice cuticles directly. Normally penetrate rice cuticles directly instead of entering through natural opening. And Oprosorium formation is initiated by cessation of the tip growth and germ tube attached to hydrophobic surface. The germ tube tips then begin to bend and swell. This process known as germ tube tip deformation or hooking. Okay, after after this Aposoria method, the Aposoria will progress through the maturation process that involve many structural modification and physiological activity, such as the position of additional cell wall layer, mineralization, and tube coordination. Also, conidian and germ tube cell usually collapse when Aposoria matured. After Aposoria matured, this Aposoria will do penetration. Okay, this penetration we do by matured Aposoria. So this stage we call Aposoria. Okay, on form, matured Aposoria develop penetration back to the base, the host surface, and enter cell of the leaf epidermis. Okay, this the penetration pad is usually much narrower than somatic hyphae and germ tubes. Okay, at early stage of penetration, the pad cytoplasm contains a few apical vesicles and consists primarily of a zone of expression that is continuous with a similar region in the Aposoria. And however, apical vesicle observed in elongate penetration pad at later penetration stage, and the penetration pad contains high concentration of active filament, and also actin and cytoskeletal element also may be involved in the selection of pad emergence site and reestablishment of polarized. After growth, this penetration pad will go to invasive growth. Okay, after penetrating the host cell, the penetration pad develop into an unbranched thickness primary fashion hyphae or vesicle, which then further differentiates into branch bulbous lobe secondary infectious hyphae. The secondary, okay, for this part, mycelium also reproduce in the cell. So the secondary infection hyphae, often simply known as infection hyphae, are morphologically distinct from germ tube and somatic hyphae. Okay, for this part, from the invasive growth, the effect for invasive growth will be produce lesion, means the injury for the surface of the plant. Okay, and this part we call it lesion. And for the long period, if the high humidity and high temperature, the disease development successfully. Also, the blast disease is also favored by excessive nitrogen fertilization, aerobic soil, and root stress. So, also in invasive growth, we will produce mycelium, and the mycelium will produce the conidia. So, the conidia will rotate the cycle and spread to the other plants. So, that's all for me. Now I will pass to the Dinesh. Thank you.
Thank you, Fikri. Then I'll continue with the field symptoms of Machnophotoi grisi. The symptom of rice blast which caused by Machnophotoi grisi on paddy include lesion that can be found on all parts of plant, including leaf, leaf collar, neck, pedicels, and seeds. From the seedling stage through plant maturity, rice blast progress through several phases, starting with leaf blast, followed by collar, pedicel, and node blast. Firstly, the symptoms on rice leaves. On susceptible rice cultivars, lesion may initially appear grey-green on younger leaves and water soaked with a darker green border and expand rapidly to several centimeters in length. Then older lesions appear as white-grey, encircled by a red-brown and shaped as a diamond. On resistant cultivars, lesions often remain small in size, which is 1 mm to 2 mm, and brown to dark brown in color. Lesions assemble and may kill the whole leaf under favorable condition. Then the symptoms on rice collar. The lesions are present at the copulation of leaf blade and leaf sheath. Collar infection may kill the whole leaf and can spread to and around the sheath a few millimeters later when the plant is damaged in the collar region. Infection on rice collar cause hinder the filling of grains, resulting in reduced quality and weight of grains. Collar infection can kill the entire leaf and may extend a few millimeters into and around the sheath. Followed by symptom on rice neck and penicills. Neck blast is the most damaging stage and may occur without significant damaging of the leaves. The signs are more extreme in case of neck blast, marked by the infection at the base of the penicil and it start rotting. This infection lead to a condition called rotten neck or neck blast. Infection produce triangular purplish lesion and expanding lesion on both sides of the neck node, symptoms that are very harmful to the development of grain. Younger nodes which attack from the neck create white penicills in color. This causes failure of seed to fill or causing the entire penicil to fall over as if rotted. The lesions are often grey-brown discoloration of the branches of the penicil, and over time the branches may break at the lesion. Finally, the symptoms on rice seeds. Seeds are not produced when penicil become infected and this condition called blanking. Symptoms of rice blast on seeds themselves consist of brown spots, blotches and diamond-shaped lesions. Thank you. Then Mukmin will continue his presentation with the diagnosis test.
Okay, thank you Dinesh for his presentation. Okay, right now I will continue with the next part, that is diagnostic test for Magnaporte gricia. For Magnaporte gricia, we commonly use polymers chain reaction, or in the name is PCR. What is PCR? PCR, stated by Delido et al. 1993, is that PCR is the melding of technique for repeated rounds of DNA synthesis with discovery of a thermostable DNA polymers, has given scientists the very powerful technique known as polymers. Okay, now, why does we use PCR for this disease? Because PCR is a method that commonly used to identify pathogens such as fungi. It also can be used to identify Magnaporte gricia that causing rice blast disease. For PCR methods, there's a 4 key ingredient to do this method. First key ingredient is Taq polymers, then primers, thirdly template DNA, and lastly nucleotides, or in another name DNA building block. Okay, what is the basic step for polymers chain reaction? Okay, firstly, the pathogens, or in this part we use Magnaporte gricia, were isolated. Then it were rutilin culture on potato dicrous agar, PDA. The plates were placed under lights and dark regimes of 10 and 14 hours, respectively at 25 degrees Celsius for 5 days. After that, the DNA of Magnaporte gricia extracted from the culture. Then we will continue with the PCR, where the Magnaporte gricia DNA are put through repeated cycle of heat and cooling to allow the DNA to synthesize. There's a 4 phase or 4 step for the PCR. Firstly, the denaturation. Denaturation is when in temperature 96 degrees Celsius, the high heat separates the DNA strands to produce single-stranded template DNA for the next step or the next phase. Okay, for the next phase is annealing. Annealing is where in temperature of 55 to 65 degrees Celsius, the temperature sets to low to cool down the reaction, so the primers can bind their complementary sequence with a single-stranded template DNA. And for the last step, extension. In temperature 75 degrees Celsius, the increase of temperature will cause stuck polymers to extend the primers. Then it will synthesizing new DNA strands. Then this cycle will be repeated for few times, around 25 to 35 times. Okay, next is a diagram that show how the three phase occur or the three step occur. As we can see that during the denature, it produces a single strand of DNA. Then there's annealing, they bind together with the primers, and the extension is synthesize new DNA strands. You can see the arrow of the blue. Okay, that's all for my parts. Next, I will pass it to Shamsiva for management control for Rice Plus. Thank you.
So, next I will tell about the management option available for the rice blast fungus, or known as Manaforte gracia. I have found 5 management options, which are cultural method, resistant varieties, biological method, chemical, and lastly use of botanical. So the first management option is cultural method. For this method, we can apply high amount of silicon to reduce the frequency of episterial penetration by the blast fungus. For example, results of work by Hasaka et al. In year 2008 have suggested that the proportion of the episterial penetration after 111 hours decreased 5-fold, and the proportion of penetration that developed to lesion decreased 2.5-fold when the amount of silicon application increased from 0 to 0.8 gram per plant. The next management is resistant varieties. Majority of the farmers nowadays decided to plant blast resistant varieties of rice. There are about 100 quantitative blast resistant R genes and more than 350 quantitative trait QTLS have been identified in rice, and 19 R genes have been successfully cloned and characterized. So R gene is a dominant gene, and it is effective in preventing disease by blast fungus. So we can pyramiding the R genes in rice cultivars as the strategies to overcome the relatively small effects of single R genes and further improve the resistance to rice blast. The third point is biological control. Biocontrol of rice blast mainly relies on the use of antagonistic bacteria and induction of host resistance. For example, strain of PF7 to 14 of Pseudomonas fluorescens was found promising in reducing blast disease significantly in field experiment by Gnanamanikam and Mew in year 1992. The result of this experiment show that strains designated PF4 to 15 and PF7 to 14 of P fluorescent accounted for 59 and 47 leaf blast reduction respectively. The suppression of disease by Pseudomonas fluorescens was likely due to antifungal antibiotics AFA, which afforded 70 to 100 inhibition in conidial germination of blast fungus at higher concentration, which is 1.0 ppm. The next point is chemical control. In this method, we can use systemic fungicides with different mode of action to control rice blast disease. For your information, using fungicide with different mode of action in the rotation or in combination ensure the better efficacy of fungicide and also lowers the emergence of fungicide resistant strains. Systemic fungicides have efficacy after the fungus has penetrated the plant for a limited time, example 24 to 72 hours, depending on the fungicide, disease and the rate used. How it works? Fungicide will absorb by the plant cuticle and underlying tissue and can act by killing spores and hyphae as well as incipient infection where the fungus has penetrated the plant surface. The last method is use of botanical. For example, combination of bale and tulsi leaf extract with chemical is also one of the effective methods of controlling leaf blast. Work done by Ama Bioha in year 2000 found that commercial botanical products with principal ingredient as azadirachtin is effective in blast management. The combination of botanicals item and chemical can play a significant role in preventing germination of blast fungal spore. That means if the germination of blast fungal spore are being prevented, so the development of disease also will be interrupted. That's all for the management option available. Thank you.
Finally, this is the references that we use to collect data about the Machnophothic That's all from us. Thank you.